A newly discovered bacterial enzyme pair can convert A type to O type blood, which means that many more of us may be universal blood donors in the future. Welcome to Microbial Minutes, ASM's update on what's hot in the microbial sciences, the You're Just My Type edition. I'm Julie Wolf, and we'll be discussing a paper that was recently published in Nature Microbiology. This paper shows that a bacterial enzymatic reaction can convert A type into O type blood. This is significant because blood is, of course, used in hospitals and healthcare facilities around the world. Every year, there are over 100 million pints of blood which are donated, and in the US alone, over 16,000 liters of donated blood are used every day. Now, blood is typed and must be matched so that the donor and recipient are matched, and the recipient does not have an immunological reaction against some of the antigens on the red blood cell surfaces that can lead to lysis of those red blood cells and a bad outcome for the patient. You may be familiar with type A, type B, and type O blood. These are the antigens, which are carbohydrate structures on the cell surface, which are indicated in this schematic from the paper on the right-hand side. Can see that the type A and type B red blood cells have type A and type B antigens, and that the type O red blood cell has what's called a type H antigen, which is the base of a type A and type B. They contain this H antigen base and then have additional carbohydrates that are added onto it to make up either the A antigen or the B antigen. So you can imagine that enzymatic conversion by removing some of these subunits would convert some of this A and B type blood into O type blood. And in fact, enzymes that can do this conversion are already known, but they're very inefficient, as we will see. What this group wanted to ask is whether there is a more efficient enzyme that could be found within a large community of microorganisms, and in this case, they will be interrogating the gut microbiome to see if they can find a better, more efficient enzyme. On the next slide, we'll see what they found. They looked at the metagenomic contents, meaning all of the DNA content of the gut microbiome, and they took that and converted it into phosmids, which are large plasmid-like pieces of DNA that contained random pieces of the metagenomic contents. So various genes would be in these phosmids and expressed within the E. coli, and they could then be screened for this biochemical activity. Using these screens, they were able to identify two genes from the commensal bacterium Flavanofractor plautii. One is a deacetylase and one is a galactosaminidase, and these in tandem can work to convert that A type into the B type blood. And the proof was really in the pudding when they tested it on red blood cells, which I'm showing you on the right-hand side. So here they're looking at A-type red blood cells, and they're analyzing samples with a technique called FACS. So they're testing for how much of the A antigen is present on the x-axis and how much of that H antigen, which represents the type O, on the y-axis. And let's look first at that panel B. You can see in the first graph of panel B that most of the sample is found along the x-axis, further to the right on the x-axis, indicating that type A, they have that A antigen red blood cells, and that very few are in that boxed off area. That the red boxed area represent the type O blood cells. This is using one of those enzymes that I told you is already known that can convert, an enzyme called GH109 from Elizabeth Kingia meningosecticum, which has the highest specific activity for A antigen cleavage and yet is still very ineffective, as we can see. It's not at this lower concentration of 5 micrograms per mil. It converts basically none of the A antigen into the H antigen, and it's only at the highest concentration tested here, 50 micrograms per mil, plus the addition of dextran as a stabilizer, that they're able to see full conversion of that type A red blood cell population into the type O, at least as indicated as having the antigen H. However, now we can look at panel A and we see the combination of the two enzymes from Flavanofractor, and we see that at the lowest concentration tested here, the 5 micrograms per mil, that the entire population has been converted from type A antigen into the antigen H, indicating that they have been converted into this more universally accepted carbohydrate that decorates the exterior of those red blood cells. So this is a big deal because of the efficiency and the efficacy of these enzymes, which means that there would be less enzyme required, making this a more economically applicable enzymatic pair than the previously described enzymes. So this was picked up by a number of organizations, including the tech publication Silicon Valley, who featured a quote from senior scientist Stephen Withers. Stephen Withers said that two concerns that one would have is one, that we don't completely remove the A antigen, or two, that we are causing some other change on the red blood cell surface, though we have no reason to think that at the moment. And I'm sure that they will follow this up with studies in vivo before doing any application with by partnering with blood banks. This story was also featured in Science, where a scientist who works with blood banks and is unaffiliated with the study said that the findings are very promising in terms of their practical utility. After all, a third of the blood that is donated in the United States is type A, with the other 2/3 being either type O or type B. And converting this type A into the type O would effectively double the pool of universal donor blood. If you want to hear more from the authors themselves, the first author, Peter Rothfeld, wrote a piece that is published in the Nature Microbiology community, where he not only describes the research, but also indicates that he is excited for future biochemical discoveries that may be found within the genomic contents of the gut microbiome. So today we've heard that enzymes from the human gut microbiome can convert A into O-type blood. For more updates on microbial sciences, make sure that you subscribe to ASM's YouTube channel. I'd like to thank you for listening and thank Ray Ortega for production. I'm Julie Wolfe, and I'll be with you next time on Microbial Minutes.